a couple things. First off, open labs start this week. Okay, what is open lab? It's unstructured time in the lab. It's going to start to be really useful to you after you start skeletal material, muscles, cadavers, all of that stuff. Some things are really difficult to understand 2D, right? You actually need some kind of 3D. You need to touch it. So let me tell you where those resources are as times start to get stressful, which will begin next week, although a couple of the labs will actually start on axial skeleton this week. So where are these resources? So open labs, first off, right here, look at the times. We just got the times worked out this weekend, so they're different than they used to be. There are more of them. Please note that early in the damn morning on Tuesdays, 6:30 to 8:30, there's an open lab. Wednesdays, 5 to 7, this is all Redwood. And on Saturdays, 1 to 3, there is an open lab, okay? There will always be at least one instructor and anywhere between 1 and 4 lab aides there during the open lab times. And then on Jordan campus, there are 2 as well. In addition, when possible, when I can get lab instructors to agree to it before the lab practicals, I'll try to make a few more hours that week. So open labs are really useful, especially if you are struggling in lab. And FYI, in anatomy, more than in any other class, lab can tank you, right? If you let it get away from you, lab is really difficult. You can't make it up. You should never, ever, ever miss lab. There is no substitute for the time that you can have with those cadavers and specimens. Okay, let me show you a couple more things before we move on. The other places that you can find a lot of the material, not cadavers, but a lot of the material. Here in this library, they have skeletal material, and they have a lot of models, okay? On Redwood campus, I bought $30,000 worth of models for the STEM Center last semester. So they are, do you know where the STEM Center is? It's in the SI building on the first floor. They've got tons of anatomy models. In addition, there are also anatomy tutors there in the STEM Center. In addition, the library at Redwood has some models. They're not as good as the ones in the STEM Center because I just bought the STEM Center ones. And if you're a Jordan person, the Jordan campus also has some models in their library as well. So there's lots of resources for you. In addition, please note that I put a ton of crap up here on the resources. So if you go resources, study tools right here. Come on, you can do it. Okay, so there's a Quizlet for anatomy. There are printable flashcards for histology. There's a whole bunch of the models that are labeled. All of that stuff is there. So as you start to feel like you're going under, first off, make sure that you come see me if you know that you're putting in the time and still not doing well. If you're not putting in the time, then that's the first thing you're going to have to do. Anatomy takes a ridiculous amount of time. But it's actually not intellectually that challenging. It's really not like learning theoretical physics in which you're like, I don't even understand any of the nouns that you just used or any of the verbs. Right? No, anatomy is not like that. It's just an overwhelming amount of information. Okay, so are there any questions that came up since the last time I saw you guys? Yeah, go ahead.
Are you recording?
Yes, I am. Mm-hmm. Okay, so we are going to do histology today. Okay, you guys started histology in lab last week, and you'll finish it this week. Okay, before we get into histology specifically, let's broaden your definition of a tissue. In classes before now, you probably defined a tissue as something like, I don't know, cells functioning together. But it's not just the cells, it's also the stuff that's between the cells. Because, for instance, blood is a tissue. I know it sounds wacky, but it is. But bone is a tissue too, and they're both connective tissues. And the biggest difference between them is not the cells, but the stuff that's between the cells, right? Because with blood, what's between the cells is about 90% water, which makes it a fluid tissue. But with bone, what's between the cells is calcium crystals and protein, which makes it a solid tissue. So it's not just the cells, it's also the stuff between the cells. The stuff between the cells is collectively called extracellular material or extracellular matrix. Yeah? That makes sense? Okay, so we'll talk about the matrix. It varies really widely in how much you have and then what's in it. Of course, we know the definition of histology, right? And now let's just talk about the 4 basic categories of tissues and where you could find these. We're going to do epithelial tissue first today. So And then there's also connective muscle and nervous tissue. So let's do the easy ones first. Where are you going to find nervous tissue? Yes, nerves, brain, spinal cord, right? So brain, spinal cord, tons of it, right? And then the wiring that's going to and from your organs, muscles, and all that stuff. So that's nervous tissue. Muscle tissue. Where are you going to find muscle tissue? Skeletal muscles, of course, right? But you wiggle other places that are not skeletal muscles. So where else? What? Yeah, cardiac muscle. And then any place inside the body that you need to wiggle or push something around that's not the heart, you're going to use smooth muscle. So the walls of almost every tube that you have, the walls of your digestive and urinary system, diaphragm skeletal muscle. So you are going to have 3 different categories of muscle tissue. Those are pretty easy because there's only 3, right? And then epithelial and connective are probably the challenging ones. I think epithelial is a little easier. I think there are 10 epithelial tissues, maybe 12, I can't remember, that you learn. FYI, you are not learning every tissue in the body. You are learning the most common tissues in the body. So please don't ever be surprised if you run into a tissue that you haven't learned. I'm not going to test you off of those. They do exist, though. So here's where you find epithelial tissue. My anatomy professor told me that you find epithelial tissue anywhere you can stick your finger, if your finger could be infinitely small and infinitely long, right? So epithelial tissue, eyeball epithelial tissue, up my nose epithelial tissue, in my ear, but also down my digestive tract and all the way out the other end, into every single pore that I have, up the reproductive tract, up the urogenital tract. That's all epithelial tissue, right? It's found at the boundaries of you and the external environment, but it's also found lining all of your internal surfaces. So if I was looking at what was going to be lining the stomach, it would be epithelial tissue. If I was looking, and I had to guess, the superficial surface of a serous membrane would probably be epithelial tissue. Exactly, right? Okay, lining every single blood vessel is epithelial tissue. So basically, if you come to a boundary, and there was tissue and then there wasn't, epithelial tissue, right? In addition, it's also going to form glands, but we'll do glandular epithelium a little differently. Okay, so that's epithelial tissue. And then what's connective tissue? Well, connective tissue is weird because it's like this catch-all. It doesn't really have like a consistent prediction of what's going to be connective tissue, but it's not nervous, it's not muscular, and it ain't at the surface of anything unless you broke something, right? So what's connective tissue? If you needed packing material between one tissue and another, it's always going to be connected, right? You need to strap something to something else, it's going to be connected. If you need something to resist gravity, like cartilage and bone, it's going to be connective tissue, right? So why, and also weird stuff like blood is a connective tissue. Why such weird, diverse tissues all go into one category? It has to do with embryology more than adult tissues. They are all derived from the same embryonic tissue, so they're all considered connective tissue. I'll be able to organize them for you so that you'll understand them. But that's, I think connective tissue is probably the hardest. A, there are the most tissues, and B, there's not a naming convention. Like with epithelial tissue, it's blank, 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 generally simple, squamous epithelium, right? Connective tissue, so you just name them whatever the hell they wanted to, right? And there's no convention, right? But I will help you at least recognize why they were called that, even though there isn't a prediction that they would have been called that. Okay, so we are going to talk about organs too, of course. So if you have an organ, your prediction would probably be that any organ would have at least one of each of the 4 categories of tissues. So I want you to think about the heart, because everybody knows what the heart looks like. And even without knowledge, I want you to try to predict what category of those 4 tissues would be on the surface of the heart. Epithelial tissue. And then before I run into the wiggly stuff, I need packing material between them. That would be connective, and then I would run into what category? Muscle, right? But now I'm going to go all the way to the lumen of the chamber of the heart, right? All the way past the muscle, and I'm working my way toward the inner surface. On the other side of the muscle, I should find what? Before epithelium. Connective tissue, right?
So it goes
right?
Like backwards.
Yeah, exactly. And then I'm going to run into what lining the inner surface of the heart? Epithelial. Okay, but I haven't hooked up the nervous tissue yet. But if I want to be able to control the rate or strength of cardiac muscle contraction, I have to wire the nervous system to it, right? So notice you've got at least one instance of every kind of tissue in that organ. And you're going to see that really common. That's not always one, but it's at least one. Okay, that makes sense? All right, so now we are going to move to epithelial tissues. Okay, so Referring to these, you can either call them epithelial tissues, noun, adjective, noun, or you can call them epithelia, which is just a noun. So it can be an epithelium, which is singular, or epithelia, which is plural. So for instance, I think you are learning 10 epithelial tissues, or you're learning 10 epithelia. Does that make sense? Okay. All right, so since epithelial tissues generally are at the surface, they have surface functions, right? And the surface functions are varied. I'm not telling you every epithelium does each of those things, but you're going to find that at the surface is a good place to have protective functions. But let's just have a prediction here. If I have an epithelium on the surface and it's very, very thin, is it going to be very protective? No. If it's very thick, it's more likely to be protected, but it's less likely to be good at transport, like absorption, filtration, or secretion, right? So the thickness of the epithelium can imply to you its function, right? So transport is difficult across thick surfaces. It's really easy across thin surfaces, right? So protection, secretion, since glands are made of epithelium, then they are secretory structures. And then absorption and filtration. So if I want to get anything into or out of the body, I have to cross an epithelial tissue, don't I? Right? So absorption is bringing things in, and I have to cross the epithelium, for instance, lining my small intestine in order to do that. If I were trying to do filtration, that would happen primarily across the epithelium of the kidneys, right? And then also capillaries, good. And then excretion. Does anybody know the difference between secretion and excretion? It's fine if you don't. They're both going across, and they're both usually out, but not necessarily outside. Has to do with what you're secreting. What? Usually, yeah. And it's primarily got to do with whether you want to keep it or whether you want to get rid of it. I would, for instance, secrete a hormone, yeah, because I made it on purpose. I wanted to keep it, and I would excrete a waste product. Does that make sense? Sweat. Yeah, like sweat. Mm-hmm. That makes sense. Okay. So secretion and excretion, and then last but not least, because it's at the boundaries, it's a really, really good place to do sensation, right? Really good place to sort of gather information about the world around you. Okay, so that is not meant to be a comprehensive list of what epithelial tissues do, just sort of a tour. Okay, so where do you find it? Like I said, you find it anywhere you can stick your finger, covering body surfaces, inner and outer, lining body cavities, and then also forming glands. All of your glands are made of epithelial tissues, even if they're not on the surface. So that's like an exception. So generally, when we're talking about epithelium, we're talking about covering and lining the surface epithelium, and then glands. Okay, so let's look at some characteristics of epithelial tissues. So although it seems counterintuitive, epithelial tissues are usually avascular. You may have never heard it, but I'm sure you can figure out what that word means. What does it mean? No blood vessels within the tissue. So guys, here's the epithelial tissue. Please note that I do not have a blood vessel in there. I don't see a cross-section or a long section of a blood vessel within that tissue. I do see it below that tissue. Yeah? Okay, so they are usually avascular, which seems kind of counterintuitive because the tissues are also really active in cell division and regeneration because they're at the surfaces and they're going to need to be, right? Friction damage, right? So how do you manage both of those things? Really active in cell division and regeneration, which implies need for nutrients and oxygen, right? But no blood supply. Well, what happens is even the thickest epithelium is microscopically thin and has a really rich blood supply beneath it, right? So as long as you've got a really rich blood supply beneath you and you're really, really thin, right, you can actually diffuse oxygen and nutrients up and diffuse waste products and carbon dioxide down, as long as the tissue underneath is really richly vascularized. Does that make sense? So it's kind of like make a great jelly sandwich, accidentally sit it in a puddle on your counter, right? And then give yourself 20 minutes before you come back. Is the top slice of bread going to be purple? Yeah, it is, right? Is the puddle going to be purple? Yeah. But if you put 15 sandwiches, is the top slice of bread going to be purple? No, this only works well, diffusion only works well over microscopic distances, right? So as your epithelial tissues get thicker and thicker and thicker, this transport mechanism isn't as effective, okay? But still, even the thickest epithelial tissues are still thinner than, for instance, cartilage. They're way thinner than cartilage. Okay, so they're usually avascular, but although it doesn't show in this picture, they are usually highly innervated. And what do I mean by highly innervated? Even if you haven't heard that word before, what does it tell you?
There's a lot of nerves in there.
Lots of nerves going to and from. Lots of sensory capacity, lots of ability to tell changes in temperature, changes in pressure, pain, itches, all of that kind of stuff occurs at the surfaces. So abundant nerve endings, and epithelial tissue is capable of rapid cell division and regeneration, which makes sense because it's at the boundaries of things. It also implies something about epithelial tissue that's important. Epithelial tissue, compared to the other four categories of tissue, other three, sorry, has the most rapid rate of cell division and regeneration, which gives you some prediction about its predisposition to cancer compared to other tissues. The tissues that are most likely to become cancerous are your epithelial tissue. So just think about cancer for a second. Think about the most common kinds of cancer that you encounter. Skin cancer, right? Breast cancer, that's glandular epithelium.
Colon.
Lung cancer, colon is the lining of the colon, which is epithelial tissue, right? Ovarian cancer occurs at the surface of the ovary, which is epithelial tissue. Lung cancer occurs on the lining of the lungs. So what you really, really want to do is, of course, protect all of your tissues from carcinogens, but specifically protect your epithelial tissue from carcinogens. They already have a rapid rate of cell division and regeneration, and then they get hit with a mutation, right? Then it's really easy to lose control of cell division. Does that make sense to you guys? So even glands, by the way, thyroid cancer, right? That's pancreatic cancer. This is glandular tissue, glandular epithelium. Okay, so the other thing that makes you able to recognize an epithelial tissue really easily is that there is no room for matrix between the cells. They're usually smacked right up against one another like fence posts, right? Or like fence pickets. So let's look at a couple of tissues, okay? Do you see this, right? They're right next to one another. There's no room for blood vessels. There's no room for matrix. There's no room for anything between them, right? So they are smashed right up against one another. Contrast that, look here. Contrast that. This is epithelial tissue. Look how close the cells are to one another. This is connective tissue. It has lots of room between the cells, and there's going to be stuff that you'll have to identify eventually between the cells. That makes sense? Okay, and then probably the most useful thing to understand about epithelial tissues is that the vast majority of them, since they are on surfaces, will have a tissue underneath one side and no tissue on the other side. They call that polarity, meaning that it has a top side and a bottom side, even if you turn it upside down. So we can label the two different surfaces. So this is the apical or apical surface, as in top or apex. And this is the basal surface. The basal surface is going to have stuff underneath it, and the apical surface is not. So if you see a free surface with a normal epithelial tissue, not something that was broken, it is telling you that this is actually found at the surfaces of things. It doesn't tell you whether it's inner or outer, but it's found at the surfaces of things. So this is called polarity. It means that the two sides are different. Like in chemistry, polarity was like positive and negative side. This just means that there is like a top and a bottom, which please note, this connective tissue right here doesn't have a top and a bottom, right? Because it's, right? Does that make sense to you guys? Okay. Now, this one does, but note to self that the apical surface can be upside down if it damn well pleases, right? Because there isn't really a right side up in your body, right? Okay. So this is the apical surface and that's the basal surface. Good? Okay. And then, how does it stay stuck to the tissue underneath it? The way that it stays stuck to the tissue underneath it is that it uses a basement membrane. Here's what the basement membrane is. Have you ever played with like a two-part adhesive that isn't sticky until both of them come together? Okay, so imagine that the basement membrane, what it is secretions from the epithelial tissue that combine with secretions from the connective tissue. And when you stick them together, they form an adhesive. It's proteins and sugars primarily. An adhesive that sticks the epithelial tissue to the deeper tissue. It's right there, okay? So it is acellular. It doesn't have cells in it. It was made by cells. It was secreted by cells, right? So it's called the basement membrane, and it keeps it stuck to the tissue underneath it. It's like if you've ever tiled a floor on top of the subfloor before you put the tiles down, you put down the adhesive, right? And then we'll talk about the grout, which is between the tiles. We'll talk about that later. Okay, so this is the basement membrane. So let me show you something. Okay, so now let's make sure we can use this terminology correctly. What surface of the cell is this? Apical. What surface of the cell is this? Basal. What structure is this? Basement membrane. Again, it's my job to write a clear question, and it's your job to answer it, right? So if I say, what surface is this? It's the basal surface, but what structure is this? It's the basement membrane. Yeah? So would both of those be choices on a multiple choice question? Yeah, they would, right? Because I need you to be specific about what you're answering. That makes sense? So that's the basement membrane. Now, basement membrane doesn't always stain beautifully like that. And usually I only ask it when it stains beautifully, but FYI, it has to be there. Otherwise the tissue would not stay adhered, right? So even though you can't see the basement membrane in all histology slides, if there's an epithelium, it is there. Good? Okay. If I wanted you to label it, I'd try to find a good slide where it was actually stained well. Okay? All right, so the other thing, one last little tidbit, is that sometimes epithelial tissues, in addition to having the basement membrane, they have little spot welds, or imagine that now what I've got is my tiles, right, and I'm sticking them onto the adhesive, which is the basement membrane. The subfloor beneath is the connective tissue, right? But I really, really want it to be super strong, so I pop a rivet through every few cells, not every one. And that actually is called a hemidesmosome. What it is it's proteins extending from the cytoskeleton of the epithelial cell into the basement membrane, right? That makes sense? It just makes it stronger. So it's called a hemidesmosome. Okay, all right, so now, I know that you know this, but I want you to think about it for a second. Just by looking at this drawing, I want you to tell me what about this drawing immediately tells you that it's an epithelial tissue other than the label. Go ahead. So it's stuck to the basement membrane, and they're what together? Really close. Okay, what else? There's other things too. That could be cilia, or it's probably cilia, but we haven't learned about that yet. So what else? Tells you it's an epithelial tissue.
Comes to the surface.
Yeah, it's got a free surface, right? That makes sense? Do you see anything between the cells? No, no really visible matrix. So what I really want to make sure that you get today as we go through the epithelial tissues is instead of rote memorization of each epithelial tissue, every single time you see a tissue, you can immediately tell that it's an epithelium, right? And then you start to figure out by process of elimination which of the epithelial tissues is it. First thing is you should always clue in that it's an epithelium, and we'll work on that as we go through. So you should, don't try to do it right now, you should of course be able to identify that as an epithelial tissue. You should be able to name the apical surface, the basal surface, and the basement membrane very easily, right? On that? Okay, all right, so now we're gonna start naming and classifying all of the epithelial tissues. I don't think epithelial tissues are super hard after you get used to them. Okay, so epithelial tissues have a naming convention that they use most of the time, which is better than nothing, okay? And the naming convention is three-name naming. So when you name an epithelial tissue, it's blank, blank epithelium. Never leave off the epithelium because you give two adjectives without a noun. Simple, squamous epithelium, right? Stratified cuboidal epithelium, okay? That works most of the time. So what are the other two names? The first one, simple, refers to how many layers of cells do I run into from the apical surface to the basement membrane?
One.
If it's one, it's simple, right? If it's more than one, it is stratified, which means layered. If it's lying to you and it looks like it's more than one, but it's not, it's pseudo-stratified. I will teach you how to tell pseudo-stratified from stratified today. Okay? That makes sense? Okay? All right, so, and then the second word has to do, with the shape. So, by the way, the best way to tell whether it's simple or stratified is to look at the nuclei. Because if it's really, really layered, you should see nuclei layering on top of one another. So simple versus stratified. And then the next word is usually the shape of the cell. You can have a squamous cell, which is squashed flat. If you look at the nuclei of a squamous cell, they are usually like the yolk of a fried egg, really squashed flat. Does that make sense? Okay. Squamous means flat. If it's cuboidal, then the cell is about as wide as it is tall, and usually the nuclei are nice and oval. Okay? And if it's columnar, it is taller than it is wide, and usually, not always, but usually the nucleus is oval, and usually it's close to the basal surface. Does that make sense? Okay, so usually can help because you don't memorize the tissue. You look at it and say, well, this applies to me that it's this, and then I can check and see if I've got this other thing. So we're going to practice that as we go through today. Okay, so let's do the simple tissues. There are 4 of them. There's simple squamous epithelium, simple cuboidal epithelium, and 2 different simple columnar epithelium. So I want you guys to know that for me, in addition to being able to identify the tissue, you also have to tell me where it goes in the body, magic school bus-like. So you got to get me small enough that you get me there. So for instance, if we got to keratinized stratified squamous epithelium and you said skin, it's not good enough because the skin is made of more than one tissue. So how do you get me to the right place in my magic school bus?
The epidermis.
The epidermis, or the superficial surface of the skin. Or if you can't remember superficial, you still have to play magic school bus and write top surface of the skin or something, right? So you have to be specific. Because remember, every organ that you're going to run into has more than one type of tissue. So if it's lining the alveoli of the lungs and you just say lungs, it's not good enough, right? So we'll practice this as we go through, okay? Ready? Okay, so first one is simple squamous epithelium. Here is a simple squamous epithelium right here. There is not a whole hell of a lot to look at because it is the thinnest of all the thin tissues, right? It is both simple and squamous, right? From the side, it's going to look like some fried eggs, and sometimes you won't go through the yolk, right? So, is this going to be a protective tissue? No. None of the simple tissues are going to be protective. They're all going to be about transport, okay? It is really not providing any impediment to transport, so not surprisingly, you will find it lining your capillaries, right? And lining the alveoli of the lungs. What happens across the alveoli of the lungs? Oxygen and carbon dioxide transport. And you don't want any impediment to oxygen and carbon dioxide transport. Does that make sense? Okay. So this is a simple squamous epithelium. So let's look a couple more places. So you can have a simple squamous epithelium from a top view. I will never ask you simple squamous epithelium from a top view because that's memorization. Because can you tell it's simple by looking at a top view?
No.
Or squamous, right? So even though it's harder to identify from a side view, it's also more meaningful. Right here, this is simple squamous epithelium. That makes sense? And you can figure it out if you just look at it, right? You're like, oh, it's obviously an epithelium, pretty damn thin, see a couple of nuclei squashed flat, has to be simple squamous. Got it? Okay, so that's simple squamous epithelium. It's the thinnest of all the tissues. The next one is simple cuboidal epithelium. Usually pretty easy to identify if you look at this tissue right here. Can you tell it's simple? Yep. Can you tell it's cuboidal? Notice you can't always see the boundary of each cell, but you can see that they're about as wide as they are tall. Nice round nuclei. This is the cutest of the epithelial tissues. I don't know why, I just think it's adorable. And that is what kind of section, by the way? What kind of section is it? That is the lumen. Longitudinal or long section. This is the same tissue in a cross section. Does that make sense? Can you tell it's simple and cuboidal, right? Go from the apical surface, final lumen, go from the apical surface all the way down to the basement membrane, simple cuboidal. Good? Okay, so that's a simple cuboidal epithelium. Simple cuboidal epithelium is found lining these teeny tiny tubes in the kidney called kidney tubules that cleanse your blood. So it's found lining kidney tubules, and you'll run into it both in anatomy and in phys. So it's really important for cleansing your blood. Okay, and then simple columnar epithelium. There are 2 different categories of simple columnar epithelium. Okay, so here is one category of simple columnar epithelium. Before we worry about cilia versus microvilli, first question is, could you look at this tissue and tell that it's both simple and that it's columnar? Yeah? So one layer of nuclei tells me one layer of cells, right? And columnar, it's taller than it is wide. Does that make sense? So both simple and columnar. This one is also simple columnar. So look from here to here. One layer of nuclei, right? Taller than it is wide. So now let's look at the difference between the two of them and why they are different. Okay, so these two tissues are both simple columnar epithelium, but one of them is ciliated and one of them has microvilli. So let's just learn the difference between cilia and microvilli. Okay, I'm going to have two sets of columnar cells. Okay, 1, 2, 3. This one's going to go 1, 2, 3. Okay, this one is going to have little protein projections from the surface. It's going to look kind of like a minion, right? So this is cilia. Okay. This is not projections from the surface. This is a folded apical surface of the cell membrane. These are microvilli. So before we get into the memorization part, let's look at it logically. The cilia have mitochondria at the bottom of them, and they can wiggle. Okay? So cilia is going to be used, for instance, to move things around on the surface. Okay? And then microvilli is increasing surface area, okay, perhaps for absorption. So which of these two, cilia or microvilli, are you going to find in the tissue lining the small intestine? Microvilli. Microvilli, yeah. And then which of them are you going to find lining the respiratory tract, cilia or microvilli? Cilia. Does that make sense? Okay, so now let's just make sure, so conceptually now you understand the difference, so now we just have to make sure that you can tell by histology looking at them. Okay, and they're really not that hard after you get used to looking at them. You ready? Okay, so if you look at microvilli, here's what it looks like to me. It looks like somebody took a highlighter and highlighted the apical surface. Do you see that? It's really, really regular all along the surface. What the microvilli actually are, FYI, is, show you a picture of microvilli with a scanning electron micrograph. This is what they do. That's what the microvilli look like along the surface. They're super regular. It's just all folded. Here it is even closer up. That's what microvilli look like. Here is a top view of cilia versus microvilli. You can see the difference. Does that make sense? Okay, so now what does cilia look like in a histology slide? Cilia looks like bedhead in a histology slide, right? You just woke up and you have not taken care of that business, right? It's all messy. It looks really messy, and it actually looks like little fibery projections from it. So I'm going to show you guys a couple of pictures, and you tell me whether you're seeing cilia or microvilli. What's that? That's microvilli. And then what is, where is that picture? I always lose it. What is that? Cilia. Yeah, that's cilia. Does that make sense? Okay. So if you know that it's simple columnar, all you have to do is look at the apical surface to tell whether it's ciliated or straight simple. The one that's not ciliated, they just call simple columnar epithelium. The one that is ciliated simple columnar epithelium. Does that make sense? Okay. And the only two you're going to learn have either microvilli or cilia. So let's talk about body locations for each of those. So for each of those, this one you will find lining the small intestine. Primarily what it's trying to do is to increase surface area for absorption, right? That makes sense? Okay. This one, you don't find lining the respiratory tract. There is ciliated tissue, but it's not this one there. You find this one lining the fallopian tubes or oviducts. Does anybody have any idea what cilia would do inside the fallopian tubes or oviducts? What? Transport the egg. So here's a weird anatomy thing. Here's my fallopian tube and here's my ovary. They're actually not attached to one another. Okay? So when you ovulate, you actually ovulate into your peritoneal cavity. What has to happen is you have to create ciliary action to sort of get some wave in the fluid, right? It's called courting the oocyte. You court it into the fallopian tube. In addition, there's smooth muscle here that's like contracting, so you're almost like creating a suction. Does it ever fail you? Yeah, it does. You can have both fertilization and implantation outside of the reproductive tract. Like you can have an implantation on your bladder, an implantation on the intestine. You're not going to carry it to term, so anybody know what kind of pregnancy that is? Yeah, it's an ectopic pregnancy when an implant cells her. I would like to talk to someone about that design flaw. Wouldn't you? You're like, put the damn thing together. And also that means that the sperm is going all the way up, out, into your peritoneal cavity. I did not give consent for that. Did you? No, that was not part of the agreement that we made. Okay. All right, so you got your two simple columnar epithelia. All right, so now let's start doing your stratified tissues. So your stratified tissues, the way that you're going to tell that they're stratified is you're going to run into more than one layer of nuclei, right? The nucleus is a really good indication that there's a living cell there. Now, as they get thicker and thicker, they get worse and worse at transport, but they get better and better at protection. So you're going to be more likely to see things that are protective as they get super thick. And if it's maybe only 2 layers, you could still potentially transport through it. So let's talk about the 2 types of stratified squamous epithelium. Okay, so there are 2 different categories of stratified squamous epithelium. Stratified squamous epithelium that has living cells all the way to the surface. Do you see? Nuclei all the way to the surface. Okay, so when you look at it, can you tell that this is stratified first? Yeah? Okay, now why is it squamous? When these cells look squamous and these don't, it's because this tissue is named for the shape of the cells at the apical surface. Okay, so it's stratified and squamous. Does that make sense? Now, if it is stratified and squamous, but in addition to all of this, you have a whole bunch of dead cells on the surface filled with this waterproofing protein called keratin, then they are keratinized stratified squamous. So let's look. I don't love the picture in your textbook, but I'll show you some more too. Okay, so do you see I have here stratified squamous plus a bunch of dead stuff. Now how can I tell it's dead? No nuclei. Does that make sense? Okay, so these 2 tissues are really protective. One of them has to be concerned about water loss and the other one does not. Okay, so let's think about these. Moist stratified squamous and keratinized stratified squamous. Moist stratified squamous, you're going to find in places that have a decent amount of abrasion but have a source of moisture. Like the proximal and distal ends of your digestive tract would be moist stratified squamous epithelium. Good? The vagina should have some friction if you're doing it right. Okay? Moist stratified squamous epithelium. Right? Okay? They have a source of moisture, but they need some protection from abrasion. Okay? These tissues, by the way, have a really rapid rate of cell division and regeneration. When's the last time you impaled yourself on a Dorito, right? And then, like, 30 minutes later, can you feel that it happened? No, you're totally cool, right? So rapid rate of cell division and regeneration. Now, if you have likelihood of abrasion and also likelihood of water loss, you do this plus you leave a layer of dead keratinized cells. Keratin is a waterproofing protein, and this is keratinized stratified squamous epithelium. Does that make sense? So if you see a whole bunch and then you run out of nuclei, but you still got stuff on top, this is keratinized. So let's look at a couple of pictures, and you tell me which one it is. Is this moist or keratinized? That is moist. This one is moist as well. You see nuclei all the way to the top. So how about this one? That's keratinized, right? Okay, so get used to practicing with those, and let me show you one more thing. You can find it a couple of different places. This is the college that I used to teach at. They have some great histology slides. So let's look here. And they also have printable as flashcards, labeled on one side and not. So do you see? So you run into nuclei up to here, and then you do not. So that is keratinized stratified squamous. Does that make sense? Okay. So where do you find keratinized stratified squamous? More specific. Be even more specific. Superficial surface of the epidermis. It actually makes up the whole epidermis, but some people don't know that. It makes up the whole epidermis. Good. All right? Okay. Where do you find moist stratified squamous epithelium? Be more specific. Lining, yes. Lining the vagina, lining the mouth, lining the anus and rectum. Good? Okay, good. All right. So 2 other stratified tissues, well, 3 other stratified tissues, 2 that are called stratified, are, this is stratified and cuboidal. Do you see? Okay. 2 distinct layers, and the cells are about as wide as they are tall. Here's a better stratified cuboidal. Right here. This one's better, right? Stratified cuboidal, a different kind of section. But can you see that they're about as wide as they are tall, and there's at least 2 layers? That's a stratified cuboidal. This is another stratified cuboidal. Good. Now, all you really have to do, and you're like, I don't see the cube. Well, you also don't see a column, and you sure as hell don't see a squashed flat egg, right? So you have to figure it out by process of elimination, right? Okay. And how do you get good at this? Do you look at one tissue? No, you look at 25 until you start getting them all right. Okay. So that's stratified cuboidal epithelium. You find this lining ducts and glands, like the mammary gland ducts, and the glands that lead to your, the ducts that lead to your sweat glands are made of this. Okay. And then the last, the one that's name stratified, is stratified columnar epithelium. So let's look at it. Tell that you, see if you can tell that it's an epithelium. Yes? Okay. That it's stratified. Yeah? And what does it look like of the 3? More likely to be columnar, right? It looks more likely to be columnar because the cells are taller than they are wide, right? So this is stratified columnar epithelium. It's actually a relatively rare tissue. The only really place that you run into it commonly is in the male urethra. The female urethra is only about 1.5 inches long, but the male urethra, 1.5 inches long on average. But the male urethra is about 8 inches long on average because it goes all the way from the urinary bladder to the external urethral orifice at the tip of the penis. I'm not telling you that the penis is 8 inches long on average because I was not born yesterday. But the urethra, all the way from the inside to the outside, right, is about 8 inches long. And at the distal end, the distal 1/3, it changes tissue type in the male. So the females don't have this in the urethra, and males do. Okay, so this is stratified columnar epithelium. Okay, and then transitional epithelium. Transitional epithelium is a stratified tissue, but they decided not to name it that way. I don't know why the hell they did it. Okay, so transitional epithelium you find lining most of the urinary tract. So the urinary bladder, lining urinary bladder. Primary characteristic of this one is that it's stretchable. Okay, and when this is the tissue when the urinary bladder is empty. When the urinary bladder is empty, you can see that it's an epithelium and you can see that it's stratified, right? But you can't tell a whole hell of a lot about the shape of the cells. So they don't make you determine the shape of the cells. Notice that when it's not stretched, the cells at the surface are dome-shaped. Yeah? What do you think this tissue would look like if I stretched it this way?
Flatten out.
It would flatten out and would look very similar to what? Stratified squamous epithelium moist. I never ask you to stretch because it's me. Okay? So I'll always ask it empty like this. So this is called transitional epithelium. You find it lining the urinary tract. And yes, it does look just like moist stratified squamous epithelium when you stretch it. Good? Okay, so that's called transitional epithelium. And then last of your epithelial tissues is the pseudostratified. It looks like it's stratified, but it's actually not. So let's see how the hell that can occur. The way that can occur is this. If it's pseudo-stratified, and of course pseudo means false, right? What happens is the cells are all attached to the basement membrane, but they're not all the same size. So if you've got big ones and small ones, it can look like it's layered, but they're all actually the same, they're all actually attached to the basement membrane. Does that make sense? Okay. So this is a pseudo-stratified tissue. So how do you identify it since it looks like a mess? Well, really, it's easy because you only are learning 2 ciliated tissues. This one has cilia on the surface. So really, all you have to do is to be able to tell it from the one other ciliated tissue that you have. And you can do that because what's the one other ciliated tissue that you have? Yeah, it's ciliated simple columnar. So look at this real quick. And then let's go back and look at the ciliated simple columnar and see how easy it is to tell the difference between these two. So here is simple columnar, ciliated, right? So really all you have to do is tell pseudostratified from simple, and you can, right? They're the only two ciliated tissues you are going to learn. So this tissue is called ciliated, pseudostratified, columnar epithelium. I always use all 4 words. You can call it whatever it says in your textbook, but it does tell you everything you need to know. It's ciliated, it's pseudostratified. The cells are mostly columnar in shape, and of course it's an epithelium. I don't count off if you write it the way that you did in your textbook, but it helps you remember everything. So this tissue, find it again, has both cilia on the apical surface, and it also has some goblet cells. So let's talk about where you find this tissue. This tissue is the ciliated tissue that you find lining your respiratory tract. Does that make sense? So you find it lining your respiratory tract. Any idea what the cilia do lining your respiratory tract? Yeah, so basically, especially here, right, you are constantly breathing in particulates that you don't want to make it to the gas exchange surfaces of your lungs. Yes? And what happens is the cilia are constantly waving up, right? Does that make sense? To try to keep it out of the gas exchange surfaces of your lungs. And then let's talk about goblet cells. Goblet cells are for mucus production. Sometimes mucus is to make the surface more moist, but what is it for here? What is the mucus production for here in the respiratory tract? So what happens? What is the mucus doing? So you wave up, right? You wave the particulates up, and then what does the mucus do? Traps them, right? And then you go, or swallow. Nobody wants to know what you do with it, really, right? And that would kill a marriage right there. That's all I have to say, okay? You keep your mucus habits to yourself. So you, they actually call it the mucus elevator. That's sexy, right? So your respiratory tract has a mucus elevator, which is constantly waving up, and then the mucus is catching it and waving up, and then the mucus is catching it. That makes sense? So this is ciliated pseudostratified columnar epithelium. It's found lining the most portions of the respiratory tract. Okay, so we already did naming the cell shape. Good? Everybody good? Do you guys have questions? Okay, let's look at a few tissues. So let's see how confident you guys are getting with identifying the tissues. Okay, so what do you think this one is? Simple columnar epithelium. It's not a great picture, but I don't see dependable cilia, right? So it's probably just straight, simple columnar epithelium. Okay, what about, ooh, this one's hard. Okay, I don't usually ask the hard ones on quizzes, but unless it's a challenge question, and then I will. Okay, so look at the surface and see if you can figure it out. That's simple columnar epithelium too. The simple columnar epithelium without cilia has goblet cells for lubricating the surface of the intestine. Okay, let's do a couple more. You ready? Which one? Stratified squamous epithelium, but which one? Moist or keratinized? Nope. Nuclei all the way to the surface. That's moist. Good. Okay. How about this one? It's not keratinized. Here's the surface.
Columnar.
It is columnar. I agree with you. Stratified columnar epithelium. Good. That's good. Okay. Cool. So I showed you that. Did I show you that? Yeah, I did. Okay. All right. So the way that you get good at histology is to look at lots and lots and lots of images. Don't just look at the ones that you run into in lab, although those are on the PowerPoint. That's for lab, right? In addition, I've got so many that I will probably never quiz you off of one that you've seen before, because I want you to get to the point that you can identify it if you haven't seen it before, because that means that you know how to identify tissues and not that you know how to identify a tissue that you've already seen, right? But you're getting used to it, right? To the point that I'm like showing you. And what I try to do, FYI, is I try to find one that's really good for that tissue, right? And you may not have seen it before, but it may even be better than the ones that you've seen before, okay? So I try to find the best iteration. Sometimes I'll use them in lecture, and then they'll show up on your quiz, but they're almost never the ones out of your textbook. Good? Okay, so last little tidbit before we finish epithelial tissues is the epithelial tissues sometimes have apical surface features and like cilia and microvilli and goblet cells, right? And not only do you have to identify those, but they're also telling you something about functionality, right? You know, for instance, if you have cilia on the surface, then it tells you something about what the tissue is doing. So here's cilia functioning on the surface, by the way. This is in the respiratory tract. This is stained it so that you could see.
Some mammals use to prevent invasion by pathogens. This is ciliated epithelium that lines the respiratory tract. The cilia
That's the top view, your mucus elevator.
Where they are swallowed or coughed out.
So that's cilia. So if I were you, I would identify which of the tissues are going to be ciliated. Can you do it now? There's 2 ciliated tissues. I'll give you that pointer. Which 2 tissues are ciliated? So if you see cilia, you narrow it down to 2. It is ciliated simple columnar and pseudostratified ciliated columnar. Good? Okay. Now goblet cells. Let's talk about what a goblet cell is. Here's one right here, a bunch of them. Here's some right here. Goblet cells produce a protein called mucin, which when mixed with water produces mucus. Okay? And mucus can have a couple of different functions. It can like catch things, right? Or it can be lubricating. So again, when you see goblet cells, they're telling you something about the function of that tissue. So do you know which tissues have goblet cells? Do you know? That you've learned? Pseudostratified has it for catching, right? For your mucus elevator. And what other one? Simple columnar epithelium, but not the ciliated one, right? Okay, so here's simple columnar epithelium. What's it for here? Think about where this tissue is. It's in the intestine, right? It's allowing things to be lubricated as they move through your intestine, right? Does that make sense? So do you hear how the thinking goes? If you see cilia, it narrows it down to 2. If you see goblet cells, it narrows it down to a different 2, right? So this isn't rote memorization of all of these different tissues. This is logic every single time you see something, right? So, okay, and then microvilli. We actually only learned 1 tissue with microvilli. There are more than 1 tissue with microvilli. And the tissue that you learned that has microvilli is what? Simple columnar, not the ciliated one, but the other one. Right? Good? Okay, so we're going to stop there for today because we're like a few minutes ahead of the other class and I want to let them get caught up. So we will not have a quiz on Thursday. You'll have one next week, but I don't know which day yet.